Now we are on live, sir. Go ahead.
A very pleasant Sunday morning to the resource persons and very good evening to Indian participants. On behalf of Department of Pharmaceutics, Bapatla College of Pharmacy, Padmavati Mahila University, and Indian Pharmaceutical Association Education Division, it is my immense pleasure to invite all the delegates and participants who occupied this virtual classroom by maintaining a social distance of kilometers. So all are following our Indian government or WHO guidelines. So thank you all. At the outset, I wish to thank the resource persons. In spite of their tight schedule or preoccupied work, they are not doing work from home. They are doing their work at their offices. And today, of course, Sunday, but tomorrow onwards, it will be very tight schedule for them. Even in spite of their tight schedule, they accepted our invitation, and we have to appreciate their professionalism in order to share the contents to our fellow faculty members as well as the youngsters and students. On behalf of organizing committee and the participants, I wish to extend my gratitude and sincere thanks to the resource persons. Immediately after finalizing this program, I negotiated with Senior Professor Dr. Nagaraj sir for the extension of his possible cooperation. And within 1 hour, sir accepted our invitation and association from the Department of Pharmaceutics as well as Padmavati Mahila University. So I wish to extend my sincere thanks to Professor Nagaraj sir. And my thanks are also due to the IPA Education Division Chairman, my one of good friend, Dr. S. Vijayadara, who also associated with this program. And the faculty members, especially the pharmacy faculty members, always ready to show their commitment, dedication to upgrade their knowledge compared with other sectors. So I am always appreciate the pharma faculty fraternity and try to understand the situation, because every day we are not following the same time because they are having work at their offices, and evening time will be difficult because they are going to occupy their offices, and our morning hours will be midnight to them. Even then, they are ready to cooperate with us. So please try to understand the situation and try to attend these courses regularly as per the timetable, as per the schedule. And here we are having a blend of academia and as well as industrial experts to cover the different aspects related to research and regulatory guidelines. So development of various pharmaceutical dosage forms with their supporting guidelines, all will be focused during this 1 week program. And on behalf of resource persons, I will assure you that there will be a wonderful, nice session for all the participants. Definitely, there will be an opportunity to upgrade your knowledge. And with respect to student community, a skill development point of view, because I have to address more things to the students, because my learned faculty friends will be having all these ideas and awareness about these faculty development programs. And why this skill is required. This current education is not sufficient to promote the students to seek the jobs. That is why the state government as well as central government are proposing various skill development programs to improve the skills and their readiness to get jobs in pharma industries. And it is inevitable to have a skill development. So those students who got that skill development at earlier stage, they will be having more experience and they will be having more value added, so that their future will be very bright. And that is the importance of skill development. Of course, there are different types of skills like communication skills or presentation skills, professional skills, etc., etc. Of course, business skills marketing skills, etc., depending upon their interest. During this presentation, how they are going to approach the resource persons, the language, how they are going to deliver their deliberations. So, another one is presentation, the art of presentation, how they prepared their slides to reach the audience, creativity, efforts involved in preparing these slides. So that is a type of communication. And there are different types of memories, like pictorial memories, that is visualization of some symbols, some movies, etc. Next one is by studying. But compared with visual and learning, they practice by doing some experiments, that is practice schools, etc. So that will be more beneficial for the students, and that memory will be long standing compared with the learning and observation of some videos, etc. And here we can develop such kind of skills by learning the novel approaches from the eminent personalities. And in this connection, I wish to quote one example. Because of this COVID, everybody knows that today what will happen, only we can predict. Tomorrow what will happen, nobody knows. And one of the molecule, hydroxychloroquine, was not having much marketing, but suddenly it became a blockbuster. And if such kind of changes are there in the pharma industry, now in the pharma industry we are having a batch production. But if a molecule is having a large demand, then we have to go for continuous production. So how to transform this batch production to a large scale production? What sort of transformation? What sort of skills are required? The latest skills like PAT technology, that will be more beneficial. Such kind of skills are required. And I hope the students will learn such kind of things. What is stoichiometry, automation, machine learning, and artificial intelligence. So such kind of skills are required. And on behalf of the faculty and with the association of senior faculty members like Professor Nagaraj sir, definitely we will try to conduct such kind of programs in future also in order to benefit the student community. And once again, I will thank the resource persons, my fellow fraternity, teaching fraternity, and my beloved students. And on behalf of organizing committee, once again, my wholehearted welcome to all the participants. So please share your inputs after delivering these contents, so that if improvements any, definitely we will work out. Thank you once again. Now I will request the convener, Madam Ponganwali Madam, to chair the dais. Madam.
Thank you, sir. Thank you for addressing the participants. Now I would like to introduce the profile of today's resource person, Dr. Harana Kumar Vaddi. He did his B.Pharm from Annamalai University in the year 1990. He graduated M.Pharm from Jadavpur University, India, and then PhD from National University of Singapore. Later, he pursued his postdoctoral fellow program in University of Kentucky, USA in 2003 to 2004. Dr. Vaddi has several outstanding research publications in circulation in the field of transdermal drug delivery system. He has several individual and team awards for his yeoman contribution as a testimony of his research accomplishments. He presented presentations in American Association of Pharmaceutical Scientists and Controlled Release Societies. Dr. Vaddi is leading a bigger R&D team in Catalanta Pharma Solutions, USA. Prior to that, he worked in specialty and innovative multinational Novan Pharmaceuticals and Novartis, USA, in various capacities as scientist, principal scientist, and director level. We welcome you, sir. Dr. Harana, you can start the program. Start your presentation.
Thank you. Can you hear me?
Yes,
Yes,
sir.
hello.
Yes,
Oh,
sir.
can you see the slides?
Yeah,
Can
we
you
are
see the
waiting
slide?
we are waiting for your screen.
Can you see the slides?
No, sir. No.
Okay. Something wrong there.
Yes, sir. You are sharing.
Oh, you can see now?
Yes, sir. Yes, sir.
Okay. Okay. So you guys can see my slide, right? And can hear my voice. Is that right?
Yes,
Hello?
sir. Go ahead. Go ahead.
Okay. Okay, good.
Thank you, sir.
Okay. Thank you for that nice introduction about myself. And thank you for giving the opportunity to share some of my transdermal experience with the faculty, academia, and students. Um, Before I get onto the transdermal, let me give the disclaimer. These are views expressed solely mine, nothing about my previous company or the current companies or the figures copied from the internet and literature. Why I say about the previous company and current companies, the previous company is very important because I worked in a transdermal specialty company for 10 years. And before that, my transdermal experience, I did my PhD in transdermal and a postdoctoral fellowship in transdermal. While I was in the transdermal postdoctoral fellowship, I did a lot of industrial projects. So definitely I need to give this disclaimer about the transdermal topics I'm going to speak now. Hello, it's not moving. Okay. Okay. I'm going to touch the topics here: innovation in transdermal drug delivery, key considerations. Transdermal drug delivery is a huge field. It started about in 1980s, formally started in 1980s. But later on, so much of research being done and so many molecules are on the market. When I say the transdermal patch formally, informally in the traditional sense, these patches have been used in forest countries for many, many centuries. In Japan, in China, in South Korea, even today, the patches are well accepted though in those countries than any other country on the globe. When it comes to the formal industrial scientific way of developing, started in 1980s with the scopolamine as being a first patch, and later on, so much has been gradually developed from the regulatory agency perspective and also from the industrial perspective. And there were so many key considerations from the skin point of view, from the patch perspective, for the number of specific tests need to be done for this one. And regulatory agency is raising the bar for more and more requirements from the industry for the new products. And the industry also working hard to give what are the data that regulatory agencies are asking for. And at the same time, this industry is also trying to do more research to give a better product to the market. So the key consideration I'm going to discuss here mainly from the skin point of view, as I said, and patch point of view and manufacturing standpoint, and also the specific test. From the skin point of view, why skin? Why skin is a route for the drug delivery? This is going to discuss here. I'm going to discuss this one here. Skin has a lot of benefits that is giving, this is the biggest organ in the body. This is the largest organ in the body, and everyone knows that largest organ. Some of the benefits that skin is offering, actually the challenges for developing a transdermal dosage form. I will explain that one as I go. But the skin, probably this is everybody knows what are the functions of the skin. You might have studied in your bachelor or even high school. So why I'm going to go a little bit of basics here. you can appreciate when I say how it is relevant to the transdermal. It has been the largest organ in the body. That means it gives so much of area for drug application, whether it's a topical or a transdermal. It is the biggest area that gives. There are so many dosage forms, parenterals. You can inject through the skin, but you are putting something on the skin, and that is getting delivered to the blood. That is the challenge here. So it offers the largest organ, I mean the largest surface area to apply the drug. It protects the underlying tissues from injury. The protection is another challenge for the transdermal. When it protects, then you have to go across this protection barrier and release the drug. That is the challenge there. And prevents excess water loss. Again, water loss is something, when you put the patch, we are actually stopping the water loss that will have issues with the thermoregulations, irritation, sensitization, not sensitive, irritation, and some other issues. As I said, it's a temperature regulator. When you put a patch, especially if you put a big patch, most of the patches come about 30 centimeters square. But however, if you see the patches for the pain relieving or some kind of local anesthetic patches like lidocaine, those patches are bigger, like 140 centimeters square to 100 centimeters square, even 200 centimeters square. So when you stop this regulation, the water loss, there will be some kind of irritation and some other problems. It excretes some of the things we don't want to as an excreter, and also it maintains the temperature of the body. Serves as a sense organ. This is a very basic thing, and sense organ is very important because we need to keep that comfort level all the time. If the patch you apply and it causes discomfort, especially with the patches, you have anywhere from 9 hours to 7 days. The patches available for various drug molecules, especially the one you are wearing for a long time. You need to maintain the integrity of the skin as much as possible, and at the same time, you should be able to release the drug into the blood. This prevents entrance of foreign bodies, is microorganisms in foreign molecules, chemicals. That is again the challenge. Our drug is considered as a foreign molecule that should go across the skin. Serves as a seat for origin of vitamin D. So these are the different functions of the skin. Those functions are the challenge for the transdermal drug delivery or transdermal patch development or transdermal gel development. How you come across, how you overcome these challenges. At the same time, you don't play with the integrity of the skin. That's the part of the product development. Without discussing about the structure of the skin, any discussion about the transdermal delivery is incomplete. The skin, the cross-sectional of the skin, it has mainly 3 layers. One is epidermis, second one is dermis, and third one is hypodermis. Especially from the transdermal point of view, epidermis is very, very important. You see this part is an epidermis. This from here to here is an epidermis. From here to here is a dermis, and below that is a hypodermis. When it comes to the dermis and hypodermis, that is well connected with the blood vessels. So, and nerves, when the drug reaches to dermis and hypodermis, it is considered already in the systemic circulation, because the dermis and hypodermis is well connected with the blood vessels. It will take up the drug easily from there. But what is the challenging part? The epidermis. Epidermis, they are not well supplied with the blood supplies, especially the topmost layer, which I will discuss in my next slide. And the epidermis will have another 4 layers below that. The resistance at the 4 layers will be relatively less compared to the topmost layer, which is considered as stratum corneum. So for all practical reasons, we consider epidermis is the resistance layer for any drug permeation or chemical permeation. But the dermis and hypodermis, for all practical reasons, we think that that is not a barrier for the drug permeation because of the very good blood vessel supply. Again, one more thing is that the topmost layer, maybe I can discuss in my next slide too, the topmost layer is very hydrophobic. As it goes down, the hydrophobicity decreases with the increase of hydrophilicity. So when it comes to the dermis, it's almost like a hydrophilic nature. So this hydrophobicity and hydrophilicity are very important, how we choose the molecules for the transdermal delivery. Those are some of the considerations. This is a close view. Then, okay, this is the epidermis I was telling. Epidermis is mainly consists of 5 layers. The most important thing is the stratum corneum. The most important thing is the stratum corneum, that is the topmost layer, followed by the granulosum, spinosum, basilae. So among all this, as I told earlier in the previous slide, the hydrophobicity is very high in the stratum corneum. As it goes down, it decreases. On the other hand, hydrophilicity keep increasing. The stratum corneum, it is so thin layer. It is about 20 to 30 microns thin layer and very transparent. 20 to 30 microns means is very, very thin, very transparent. It's, even if you scratch your skin, you can see a little bit of white layer. That is the stratum corneum. The stratum corneum is so thin, but on the other hand, it has extraordinary resistance to any molecules to get into the body. It is also called as a dead layer or horny layer, but it's not really a dead layer. It has a very good function to how to protect the body. The stratum corneum has cells called corneocytes. The corneocytes are full with protein called keratin. And between the corneocytes, there are lipids. Those lipids are arranged in a bilayer fashion, lipid bilayer fashion. I will explain that stuff in my next slides. Below that is a stratum, granulosum, and spinosum. As I said, the hydrophilicity increases, but stratum corneum is the major barrier for drug permeation. Let me go to the next slide. I think you guys are seeing here. This is the stratum corneum, stratum granulosum, stratum spinosum, and stratum basilae. Okay, what is the composition of the epidermal cells? If you see, this is the basal layer, this is the granular layer, and this is the stratum corneum. And if you see, most of them are lipid nature. And if you have the basal layer has a lot of lipids here, granular layer has a lot of lipids, and stratum corneum, as I said, this is the layer of our interest. It has a lot of cholesterol, ceramide, fatty acids, triglycerides, hydrocarbons, and also keratin. A lot of research is going on to find out what is the structure of those ceramides, how they are arranged in a crystalline fashion, or what is the arrangement. Still, we don't have 100% understanding, but a lot of work is going on to understand the stratum corneum and how to manipulate the stratum corneum to increase the drug permeation. Go to the next slide. These are some of the animations I did not do. Okay, why we go to transdermal drug delivery? Probably I will go first. I think something is missed here. Okay. Why transdermal drug delivery? Something is missed. Anyway, I can tell. What are the advantages of the transdermal drug delivery? They are very straightforward. When you are applying on the skin, what are the advantages you are getting? The first advantage, you can get a large surface area from the top to bottom. There are different areas you can apply the skin, the transdermal patches or the transdermal gels. Second, you can have long-acting formulations. Long-acting formulations in the sense you can get anywhere up to 7 days. The longest available patch in the market is for 7 days, and the shortest is for 9 days. So you can have longest permeation. The third one is that if you take anything by oral route, you guys might be knowing that one, it will go through the first pass effect. It goes to the liver and metabolize something, and you are getting only the active, a little bit of active into the real therapeutic activity. Unless one of the metabolites also active, sometimes one of the metabolites will be equally or more active than the drug. In that case, the things are different, but in most of the cases, you get the drug will be metabolized and only the drug will be active. So if you take 100%, say for 100%, you will get only 30% of the drug actually available. I don't know, I'm just going a little bit fundamentals, but I don't know where your levels are. Thinking that I'm teaching to I'm giving a lecture to a bachelor students or little master students. So when you apply onto the skin, it directly goes into the blood, bypassing the first pass metabolism, and whatever the drug that is given that is 100% available. Therefore, you can decrease the toxic effects and you can use less amount of drug compared to the oral route. And another advantage is that there are long-acting formulations like matrix tablets or depot injections and all. The disadvantage is that once you give that dosage form for administration, because of some unexpected effects or anything, you cannot stop the treatment at the point. But on the other hand, the transdermal, just by take off the patch, you can intervene the treatment. You can stop the treatment there, unless your drug is very lipophilic with the log p value of like 4, 5. They tend to accumulate in the stratum corneum. When they accumulate, they give the depot effect. They gradually, even after removing the patch, the drug accumulated in the stratum corneum gradually releases the drug. So that is the one reason when you're running the clinical trials, you have to see how much drug is getting into the blood even after removal of the patch. Generally with the log p value about 3, 2.5, you don't have any problem. You just take off your patch, you intervene in the treatment. These are some of the advantages. If you just keep thinking what you can come up with, you can come with hundreds of reasons why we want to go with the transdermal drug delivery. However, it has such a number of benefits, but there are some limitations too. You cannot go with every drug molecule into transdermal, because obviously, the big molecules we cannot put into the transdermal. I'm talking about the passive transdermal drug delivery. There is an active transdermal drug delivery. I will touch base a little bit later. Those are being developed to use for large molecules, but at these passive patches, mainly for the small molecules, anything molecular weight below 500, only feasible. Above 500 is a little bit large molecules to get into the blood. And there is a trans-appendigeal route, which is like the hair follicle route, and they are just 1% of the total skin. Those routes have been tried, but they are not really that successful. I don't see any product in the market for trans-appendigeal route. So I'm talking about the small molecules, and the second one is and you have to have the appropriate log P values. It should not be too much hydrophobic. It should not be too much hydrophilic. Your stratum corneum accept the lipophilic drug, but when you get to the lower layers, there will be resistance for too much hydrophobic drug, and vice versa. So the log P about 2, 3 is a something like a ideal one, and the dose you cannot pump in like 500 milligram or 1000 milligram. Generally, it is accepted that generally it is said like less than 10 milligram. When I said less than 10 milligram, that is the bioavailable dose. If I give a tablet 100 milligram, 10% bioavailable, that's a 10 milligram. That is the 10 milligram I can think about my transdermal. Anything about 10 milligram, it is difficult to pump through the skin, penetrate through the skin. You can think about bigger patches, but too big are also not acceptable. There is an acceptability from the patient standpoint, and also the bigger the patch, there will be issues with the adhesive characters and all that kind of stuff. You can think like this, and you will get. I think I wrote down all that stuff, but there's somehow that particular slide missed. But those are the advantages with the transdermal, and also limitations, what you can do and what you cannot do. You need to understand a little bit of diffusion mechanism, how the drug is getting through the skin. Okay, I don't know, you guys might have studied your Fickian laws about the drug diffusion. This follows the Fickian law. The drug permeation through the skin is considered to be following a Fickian law, the first law. Then here is a little bit interesting and contrast. The Fickian first law is explained for the homogeneous membranes, and it is working very well for the homogeneous membranes, but not for the heterogeneous membranes. The skin, the epidermis, especially in a heterogeneous system, especially the stratum corneum, you have a lot of lipids, carnocytes, keratin, and it's very heterogeneous. But even for this one, the Fickian first law is the closest accurate one. There were many trials come up with the various mathematical models. Those mathematical models either not better accurate than the Fickian first model, or they're cumbersome to use. So far, I haven't seen anyone using the other mathematical models other than this Fickian first model. The Fickian first model, you're doing the calculation for the flux, how much drug is penetrating through the centimeter square per hour. This is the measurement. Here is the drug you keep here. The concentration is CD, and how much drug is penetrating here? The concentration is CR, and this is the rate of penetration, which is the flux. We say flux equal to, ignore this equation and come here. The concentration in the donor minus concentration in the receiver. This is the receiver and donor, and KD by H. KD, what is K? K is the partition coefficient between the drug patch onto the skin, especially the stratum corneum, and D is the diffusivity coefficient, how the molecule diffuses across the skin, and H is the thickness of the skin. It's not like a straightforward when it comes to it won't just go straight like this. The drug will go into any convoluted way like this. There's a lot of convoluted way, and eventually get in here. And there was a research to find out what is the longest, what is the path, the longest path in the stratum corneum. It came out to like 100 angstroms, but there is no clear agreement on that one. But H is the thickness of the skin. It is considered to be same always. And so this partition coefficient of the drug here to the skin and the diffusivity coefficient by divided by H, this is considered as a P. P as a permeability coefficient. That is one of the most important parameter. The permeability coefficient is very specific to the skin, specific to the molecule, every drug molecule. And the permeability coefficient changes when you put something to manipulate the skin, like skin enhancers. So here CD by CR, CD is the drug always you keep on the skin. CR here is when the drug gets into the skin, it immediately get into the systemic circulation. The concentration is almost insignificant here. So because of the sink condition, we say sink condition, it gets easily distributed there. So in all practical reason, we think the CD is the only one and there is no CR. So in other words, you will have PCD, that means permeability coefficient times concentration in the donor compartment. This equation is very important. You use always, anybody doing the research in transdermal, they should know this flux equal to permeability coefficient times donor concentration. Okay, here is the stratum corneum. It's a very good schematic diagram. It's copied from one of the books. Probably you can see in most of the standard books this diagram. How the drug is permeating. This is the stratum corneum. I said it's 20 to 30 microns, very thin, very transparent. I did a lot of my PhD research work with the stratum corneum. These are the corneocytes, and corneocytes has a lot of keratin in them. And these are the lipids. The lipids are arranged in a by lipid pathways. Here you can see these are the lipids. This is just magnification of this one. And this lipid pathways, you can see these are the polar head groups and non-polar chains here. So here, like polar head groups and non-polar chains. And oppositely, the conversely, you get polar head groups here and non-polar chains extending here. So the drug will be penetrating like this. Most of the drugs penetrate through the bilayer membranes. So probably the hydrophilic drugs will penetrate via the polar head groups, and lipophilic drugs will penetrate through the non-polar head chains. But a lot of research is going on, but that is the assumption, that is the, so far, whatever the research work is saying like that. But there are so many other mechanisms. Some of them I can maybe explain later, but some of the enhancers will wash away some of the lipids there, then they create some space for the drug to penetrate. Some of the enhancers decrease the melting point of these lipids, thereby it creates more semi liquid kind of environment for the drugs to penetrate easily. This is an intercellular pathway, and this is a transcellular, which means that the drug will penetrate across the corneocytes and then lipid pathways and cross corneocytes. But I don't see many drugs are going this way, transcellular route. This is a prediction, but most of the drugs they go through the intercellular route. Okay. So anybody who is doing the research in academia and industry, they have to use some kind of in vitro testing before get onto the clinicals. Clinicals are very expensive, millions of dollars, so you cannot spend that much of money there. Franz, is the name of the scientist who developed the first time this kind of static cells to do some work in vitro in the lab. This appears to be a simple, but that is not that simple. You will have a receiver compartment here and the donor compartment here. You put the skin, on the top of the skin, you put a drug, a patch or gel, whatever you put there, and you keep the receiver solution here, and you take the samples at different time points and analyze that one by the HPLC or whatever the technique, analytical technique, and you can get a plot. These are the vertical cells, also called as Franz cells. The design appears to be simple, but it's not simple, as I said. Some of the reasons being that here you have to create a sink condition here. This is the water jacket. The water jacket maintains the temperature at 37 degrees centigrade. This one, the receiver compartment, you have to create a sink condition. Sink condition means you have to maintain the concentration below 10% of its saturated solubility. If you have more than 10%, it will affect the diffusion adversely. So you have to find out what is the solubility. Generally, we use some kind of buffers, water, especially the buffers we take where the drug solubility is very high. Based on the pKa of the molecule, if it's the acid, take a little bit of higher pH. If it is a base, take a little bit of lower acidic side to keep the drug in ionized form. Obviously, ionized forms are having more solubility in the aqueous systems. So that's the theory there. And how much drug you can apply here. In the practical risk, there are two type of loadings here. There is a infinite drug loading and finite drug loading. The practical point of view, it's always the finite drug loading. Finite drug loading means you just keep the patch on the skin, and as the drug going in, you're not going to replace it. It's a finite. But infinite is that it's more like a research environment. You take a very high concentration, the concentration drug patch or solution, and you're replacing whenever after every 3-4 hours to maintain that concentration at the top. That is called infinite dosing system. The infinite dosing system is more for the, as I said, is more for the research purpose than for industrial application. Or you want to find out what is the intrinsic permeation of the drug. That is the one way you can do it. You just do infinite dosing system. You just keep adding the drug in some solution, like in mineral oil or something, and without any enhancers. Then what are the data you're getting? That is the intrinsic permeation. That means the permeation of the drug by itself, because the drug molecule, the permeation varies from drug molecule to molecule. Say, for example, molecule like nicotine, the moment you put on the skin, it just passes through the skin. Some molecules like cannabinoids, they don't really go through the skin. It has some pros and cons. The next one is flow-through cells. What are the pros and cons here? The sink conditions we create by taking out by creating high solubility here. But the biggest issue is that if you're running like a 7-day experiment or 3.5-day experiment, you cannot take the samples during the nighttime. That is the disadvantage. And the sink conditions are not well created here. Therefore, sometimes you don't get very good plots. You have to get a nonlinear regression to help that plot. This is the same thing here, but it's a side-by-side instead of top and bottom. Here is side by side, but the example, the theory, the principles, and everything are same. I think here is the flow-through cell. Flow-through cell. The principle here is that the sink condition constantly passes at particular speed, like 1 ml per an hour or something. The benefit because of this one is you can create a sink condition, excellent sink condition all the time, and you can collect the samples. It's an automated system. The samples will be collected throughout the nighttime or anytime. You just said every 2 hours collect a time point, every 3 hours. There will be drop coming here, the drop coming from here, and after 3 hours or 4 hours, then this bottle will move away, and the next empty bottle will come in here. Then it will be collected. So you are collecting over 3-4 hours sampling point. The disadvantage is that your sample is very much diluted here. The sample, you have to do some kind of sample preparation, which means that you have to concentrate your sample. And some of the things, if you take water, it's easy to analyze with HPLC, but if you're taking very, very high salt receptor solutions like Hanks buffer solutions and all that kind of stuff, have a lot of salts in them. So you need to make a sample preparation to avoid any interferences in the HPLC chromatogram with the extraneous peaks of the salts. This is the plot I was telling. When you you analyze your data and you get cumulative amount of permeated. The cumulative amount of permeated you get in microgram per centimeter square, and you plot against the time. This is for a 24-hour patch. You can get it's a beautiful, it's a very good nonlinear. And here you can see, this is the straight line. When you take the slope of this straight line from here to here, you will get the flux, which is microgram per centimeter square per hour. When you extend this one to the back, you will get the lag time. Lag time means that is the time for the drug molecule to reach steady state permeation. So, but when it comes to the transdermal, this flux is very, very important. And lag times we use, sometimes we need to decrease the lag time. Some of the molecules will have very long lag times, like up to 15 hours, 20 hours. Then we need to decrease the lag time by manipulating the skin. This is the information you get, flux and the lag time you get from here. This is about the skin. As I said, there is a large surface area of the skin that is available for the skin permeation. However, the skin permeation is not same across all parts of the body. A lot of work done, is there an effect of the age? Is there an effect of gender? Is there an effect of the area of the body? There is no clear difference in other parameters studied. However, there is a clear difference for which part of the body you're applying. Say, for example, forearm, palms are very difficult to penetrate. When it comes to the ear canal, axilla, scrotum, the permeation is very high. So whenever you are developing a patch for market, you have to, you're running in vitro studies, better to buy the same area of the skin for your in vitro studies. I have to mention about the skin, how you get the skin for the in vitro studies. We generally buy from the skin banks. There are two types. One is the cadaver human skin, that is the dead people human skin. And there is a fresh human skin from the plastic surgery. Fresh human skin is very difficult to buy and also very expensive. And we don't use quite often, only when there is a need or there is a demand for it, like the drug metabolizes by the skin enzymes. You want to see how much the drug is metabolizing, then you need to have the enzymes in a very active and fresh state. So at the time you use the fresh skin, but the cadaver human skin, you store them in -70. Generally, you should not use more than 2 years, but there are conflicting reports in the literature. Some say 6 months, some say 1 year, some say 2 years, but we don't keep more than 2 years. But when you keep for 2 years, obviously the enzymatic activity goes down. So that is the skin we cannot use for when we are expecting any skin metabolism. So whenever you say, for example, you are doing a research for birth control patch. Birth control patch generally applied below the navel. So and also young women. So better to go with 20 to 40 years old women, abdominal skin. or you are developing some hormones for PMS, postmenopausal syndrome, think about taking about 50 to 60-year-old female skin. Sometimes getting such a choosy, getting the skin is difficult because this cadaver human skin also difficult, but not as difficult as getting the fresh human skin. So you have to do the research with that one. But when you go to the clinicals, obviously you have to stick to the area where you're applying. That is the clinical work you have to do. And when you launch your product on the label, you have to mention where it should be applied. Some of the marketed products, okay. The first batch was launched in the 1980s by a company, Alza. Alza was the part of the Novartis. So Novartis actually launched this product, Scopolamine. Scopolamine is for the motion sickness. People on the sea or flights or anything for the motion sickness, they use. This one is applied behind the ear. Since then, a lot of research has been done and a lot of understanding. We have obtained and still doing a lot of work from the regulatory point, from the academic point, and from the industry standpoint to understand more and more about this drug molecules and also the skin and penetration mechanisms. Since then, there are like more than 15 molecules in the last 30-40 years we got into the market. Some of them here, and because regulatory agencies asking more and more information, not many drug molecules are coming into the market recently. However, buprenorphine was the last one about 6 years back, and the most recent one from my previous company that was launched in 2019. It's a esenapine used for the schizophrenia. That is the first patch for the schizophrenia in the world. And there are 2 more products coming in the phase 3. One is amphetamine for the ADHD and attention deficit hyperactive disorder for the children and also the adults. And there is one coming, donepezil for Alzheimer's disease. That is in the phase 3. So in last 30-40 years, we could be able to see only the 14 molecules. You can understand how difficult to get these molecules into the market. As I said, the regulatory agencies increasing the bar. They're looking for a lot of expectations. They're asking a lot of new studies that they never asked previously. Some of things like monomers testing and monomers impact on the skin permeation, a lot of new stuff they're asking. I will discuss some of the stuff later on. But I see there are 2 new molecules that are not here in this batch. Not new molecule, one molecule, esenapine, and the 2 are in the phase 3 trials. And if you see the pKas, not the log P are very important here. The log Ps are mostly about 3 to 4, from anywhere between 2 to 4. And if you see the dose are all below 10 milligram. And if you see the time period, some of them are per day, 12 hours patches, day patches, and 7-day patches. There should be 7-day patch here for birth control, but it's not here. And I think clonidine is another one for the day patch. And we did a lot of work to make it like 7-day patch. But most of them are typically day patches, 24-hour patches. But some of the patches only for 9 hours, for 9 hours of the 12 hours. Say, for example, the methylphenidate is for 12 hours patch. This is used for the ADHD. That means the children are very hyperactive. They cannot focus on anything. They just keep changing their focus. ADHD is a stimulant. So we apply this one in the morning and we have to take it off evening time. If you don't take it off, they cannot get the proper sleep because it stimulates the system. Same thing with the amphetamine, which is in phase 3. That is also for 12-hour patch. You have to take it off evening time. Otherwise, they cannot go to sleep. Some of them, most of them are per day. Some are for 7 days. For birth control patches, they're for 7 days. They also increase the compliance. Most of these 1-day patches, if you take the tablet dosage form, they will be like, you're taking like 3 tablets a day or 2 tablets a day, because you put 1 patch, that increases the compliance, drug intake compliance from the pharmacist standpoint of view. There's so much to discuss here, but another one, the molecules never made only for the transdermal. They are already existing in some other dosage form, like the tablets. For the tablets or something else. So you take those drug molecules and try to put in the transdermal drug delivery to get some benefits out of it, what you can get from the oral dosage form. From the clinical justification or business point of view, or you are getting some benefits out of it. Go to the next one. Okay. So I think up to this one, I completed the skin point of view, what is happening. Then I just move on to the, from the patch point of view, these are the things I discussed. Here again, as I said, most of the molecules are for the tablets or some other dosage form. You just use this for transdermal. And they go with a different pathway in the regulatory agency. They call here very popularly 505(b)(2). That means they're like an NDA. That means a new molecule, but it's not a new molecule because it's already existing. But you put in a different dosage form. But they get some exclusivity covered, like 4 years or 5 years. No other company can manufacture that product. So they make money during that time. Some of the products don't make really much money. The blockbusters like lidocaine patch, it was like $1 billion patch, but there are so many generic products already in the market. There is only one molecule that came directly for the transdermal, which is rotigotine. That's a new pro patch that is used for dementia in the old people. Again, there are different kind of patches. The most important patch, let me start with the most popular patch. Then I go back to the least preferred patch. This is the most, the number C. A drug in adhesive matrix is the most popular patch most of the companies make, and it's easy to make, easy to scale up. When I say easy, means relatively easy, but not that easy. It has 3 layers. The adhesive matrix, the adhesive is a PSA. We call PSA means pressure sensitive adhesive. When you put onto the skin with the pressure, it sticks there. The drug is solubilized in the adhesive here, and there is a backing. The backing will be on the top of the skin when you apply the patch. This is the liner. I say release liner. You have to remove the release liner and put the patch onto the skin. This is the most popular. Most of the drugs are available this way. And there are reservoir patches. There are some reservoir patches, but nowadays you don't see. I will tell you the reason. The reservoir you put as a solution or semi-solid kind of thing here, solution or semi-solid kind of thing, and you put as some microporous membrane here, and there is adhesive down here, and there is a liner. The biggest disadvantage is that with the reservoir patch is there is a leak. The drugs leak, especially if you put a patch, fentanyl patch. The biggest problem with the agency or from the safety perspective, if there is a leak like fentanyl, there is a chance whoever is coming in contact with the person, the loved ones like children, husband, wife, parents, whoever coming, there is a chance to pass. I mean, the come in contact, the drug come in contact with the other person, especially the drugs like fentanyl. They are very addictive drugs, and the addiction is the biggest problem in United States. There are so many deaths because of the addiction. The agency is very cautious about this and asking many companies to develop anti-abusive deterrent patches. That means how to prevent the abuse of these control drugs. But this is not well, I mean, there are some patches, but not as popular as this one. This is the other one. The adhesive matrix here, this is not adhesive. You just put in some polymer blend. This is the backing, and put an adhesive. Take one of the PSA adhesive and just cast on it. And this is the liner. There are some other ones like a control membrane. You put a control membrane between two matrices. You may make one matrix here, another matrix here, and between you put a control membrane. These kind of patches generally for longer duration. If you're thinking for 7 days, better you need to have some kind of controlling membrane. There are some birth control patch like Zulane, Arthevera. They come up with this kind of control membranes in between. But those patches are difficult to make. The scalability is a problem when it comes to manufacturing. So the first preference is always here. We start here. As I said, making a multi-laminate or multi-layer patches are difficult. Next one. So I think I touched base something about the patch here. And let me touch base something about what we are expecting here for the transdermal dosage form as a whole. We are expecting 4 important characteristics. These are like the basic thing for transdermal. The one is the permeability. You have to see whether the drug is permeating through the skin to the plasma level, to the therapeutic level. You have to do the particular PK studies, which is ADME, absorption, distribution, metabolism, and excretion, and PD, what the drug is giving to the body. Are you getting the therapeutic effect or not? You will have primary endpoints, secondary endpoints in the clinical studies, and you have to show that they are meeting those primary, secondary endpoints compared to your oral dosage form or whatever. Then the safety is very important when you put onto the skin. Safety is a systemic circulation safety, that is. Then the tolerability. Tolerability means when you put the patch on the skin, is it causing any irritation or sensitization? Irritation, if there is a severe irritation, you don't get the patch. You will not have a patch. Many drugs cause irritation. There is a software, there is a Derrick software. You put in your skin structure there. There is a scale of 0 to 5, I think. Based on where it falls, you can see this is causing severe irritation, and we cannot go forward. And it's not completely showstopper, because irritation is a dose dependent. At higher doses, it causes irritation. Then you can think about making a smaller doses or making a bigger patches to decrease the irritation. However, the sensitization is immune response. When you put the patch onto the skin and there's immune response, that is more like a showstopper. If there is immune response, you take out your patch, and second time you put a second patch, then there will be more immune response, more sensitization. But however, your dose is below the threshold value that causes the immune response, you still can think about making this molecule into the patch. And variability is the equally and very difficult issue. You have to have a patch that could stick to your skin for 24 hours, 7 days. 7 days is definitely challenging. And at the same time, when you take it off, it should not cause any irritation, any problem to the skin. Then there are some issues with the peel, tack, shear, cold flow. I can discuss them later. And stability is stability is like any other dosage forms for the tablets and capsules. However, it has its own stability issues. The main one is the addition issues. Peel, tack, shear, dissolution, and Skin permeation we don't do in the QC testing, but there is one thing these days FDA or EMA keep asking for the data, especially for the product development. But these are the 4 important and basic things to be considered for developing the patch. Some of the manufacturing formulation strategies, I don't know, I'm passing too much of time. I'm working, I don't know, this is okay. I'm already like 10:08. I don't know, somebody can tell me, is it okay if I go for another 15, 20 minutes? Okay.
Yes, you can go ahead.
Okay, another 15, 20 minutes. Okay. What are the important components? There are 2 types of components. One is the chemical components, and second one is the layers, the polymer membranes. The chemical components, one is the API, physical chemical properties, or we say pre-formulation. API, I said this is not a new API, this is not a new chemical entity. This is already in this different dosage form as a tablet or capsule or something else. You are going to get a lot of information from the literature about its solubility, it's about log P, it's about polymorphism or isomers. You get a lot of information from these APIs. But however, you have to do some kind of pre-formulation studies that is relevant to transdermal dosage form. For example, the solubility. What kind of solvents we take for this, I will explain later. Is the drug soluble in those adhesives? You are taking something to increase the solubility. Is that something dispersible in the adhesive you take? Those are some of the studies you need to do before you start experiment. And also API, whether API penetrate through the skin or not, that is something, there is a software you can use. Actually, there is an equation famously called Potts and Guy equation. You have log P value and molecular weight. You key in those molecular weight and log P values, and you will get a flux, how much flux you can get, the intrinsic flux without any enhancers or anything. That is another starting point, whether this API is good to go or not. Let me come back to this. Next one is the adhesives. The 3 most popular adhesives used for transdermal patches are silicones, acrylics, and PIB. PIB is polyisobutylene. The first patch, which was a scopolamine, was made with PIB. But I will go with the PIB later. First, let me go to the silicones. There are silicones, different kind of silicone patches are available. Materials are available with the different companies like Henkel. I don't know, that is, Henkel is very popular here. Henkel and Dow Corning. The silicones, the biggest advantage with the silicone, the adhesive property is really good. It's very good. You can keep for 7 days, no issues. You can peel it off, no issue. The silicones come with different solvents. They're like a solids in a different solvents. The solvents are ethyl acetate and heptane. Remember, these are the solvents come about, I said solids content is about 60 to 70%. So you will have about 30 to 40% of the solvents. These solvents, when you're manufacturing, you have to get rid of them because they are residual solvents. You have to maintain some, they should be below some certain level. IP, BP, or USP, they have given some limits. What are these residual solvents? They are like solvents 1, solvents 2, solvents 3, and there is how much you can have and how much you should not have based on this level 1s and 2 and 3s. This ethyl acetate and heptane, you should get rid of them when you're making a patch. The low drug solubility, that is the biggest disadvantage with the silicone. They're very good adhesive, but the disadvantage is that many of the drugs are not soluble in silicone. And amine drug compatible, this is very interesting. What happens, this is something happened with my previous company. They have a product called Detrana. The Diatrana has a silicone in matrix, and they put the drug called methylphenidate. Methylphenidate is for ADHD and is a secondary amine drug. When we put on stability after 3 months or 4 months, it became very difficult to peel off the release liner. And after doing a lot of mechanistic study, and it understood that the amine drug in the presence of silicone is making some interaction with the release liner, and release liner could not able to peel off. Then it became a issue with the agency, and agency asked many companies to check the release liner peel off, and some companies came up with the amine drug compatible. This is very particular problem with the amine drugs, especially tertiary amines and secondary amines. These polymer companies came up with silicones that are amine drug compatible to avoid that kind of problems when we are working with the amine drugs for transdermal dosage form. And second one is the acrylics. Acrylics is the another very popular one for the transdermal. The biggest advantage is this very wide variety of, you can get at least 20 different varieties of it. And another advantage is that they can be anywhere from moderate hydrophilic to lipophilic drugs you can use, because you can get hydrophilic acrylics, lipophilic acrylics. They have different functional groups: carboxyl, hydroxyl, amide. You can use carboxyl, hydroxyl a little bit on hydrophilic, and non-functional groups a little bit on hydrophobic. But you have to see there is an interaction of your drugs with the carboxyl and hydroxyl groups and amide groups. And we come across many times there was interaction with some of the drugs we used with this one, and we switched to non-functional groups. And the fourth point is that you can get a combination to get a required adhesive property, to get a required solubility of the drug. They come in a wide variety of solvents: methanol, ethanol, IPA, ethyl acetate, heptane, xylene, toluene, with the solid contents about 30 to 50%. As I said, all these are residual solvents. When you're making a patch, you have to get it to the certain level that is specified in IP or USB BP. The biggest advantage that I said is the adhesive property is not as good as silicones. When you want to go for the 7 days, silicone is the best, and but the solubility is low. But acrylics, the solubility is very good. The adhesive is low, so you can think about using mixture of acrylics and silicones. And there's a platform technology that was actually my old company, dot matrix platform technology that became off patent. Nowadays, many companies also using that. The silicones spread in the acrylic polymer as a dots. The silicones, when come into contact with the skin, they give very good adhesive property, and there is a drug exchange between the acrylics and the silicone, and silicone gets easily reached to the saturated solubility because of its low solubility. And the drugs, when the drug in the patch reaches to the saturated solubility, it pushes the drug through the skin because of the high chemical potential of the drug molecules in the patch. I think, let me tell a little bit of background for this one. Any saturated systems, they want to get rid of the saturation system to reach to the unsaturated systems. The saturated systems, the molecule will have highest thermodynamic activity. That is the maximum thermodynamic activity. That is the one principle we use when we make a patch. When we made a patch, we try to keep our drug at the maximum saturation in the patch, about 90–95%, so that the drug molecule wants to leave the patch and penetrates the skin to the blood. That is the principle we use here in the dot matrix technology. The silicones will reach to a maximum saturation, and it will push the drug through the skin because of the high thermodynamic activity. The dot matrix was really good, able to make smallest patches in the world. The smallest hormonal patch in the world was made with this dot matrix technology for hormones. It was like a 5 centimeters square, 6 centimeters square. There's like I don't know, in India still they're using the coins. If you remember, I don't think five paisa. When I was there was a 5 paisa coins. It was something like 5 paisa coin. I don't think that exists anymore in India. So it's the smallest patch that was made with the hormones using the dot matrix technology, using the combination of the acrylics and silicones, using the silicone best property adhesives, acrylic best property solubility, and increasing the permeation. Okay, polyisobutylene. As I said, the first patch scopolamine was made with polyisobutylene. The advantage with the polyisobutylene, it has a very good adhesive property. It's a non-functional. This comes in as solvents heptane. It comes as a 40% solids. The disadvantage is very highly hydrophobic. You cannot. It's very difficult to put the many molecules, drug molecules, with a little bit of on a hygroscopic side, like log P about 2 to 1. It's very difficult to put in there. And biggest disadvantage is that this polyisobutylene is very thick, and when you are doing the manufacturing, incorporating your API or you put another excipients is very, very difficult with the polyisobutylene. And but now thinking to see the possibility of combination of adhesives, polyisobutylene with the acrylics, but it's difficult. Acrylics are little on the hydrophilic sides because they don't mix very well with the PIB. Those are about the adhesives. They are the 3 most important adhesives. And come to the enhancer. Enhancer is very important. When the drug is not reaching to the therapeutic level, you have to add the enhancer. It's depending upon the drug molecule. Some drug molecules can penetrate right away. The moment it is on the skin, you don't need to do anything. It will get into your blood right away. And the best examples are nicotine. Nicotine is used for the smoking cessation, that patches, and also the amphetamine. The moment you put on the skin, it gets into. Actually, those are the toxic effects. I don't know if anybody is working with the nicotine, amphetamine, or kind of these drugs. Be careful. If you come across and if it falls on your skin, right away get into your blood and many side effects. And you have to take a lot of measures like double gloving and how you deal with those kind of medications, the drug molecules. Some, they don't really penetrate to the therapeutic level you're looking for, the blood level you're looking for. So you have to add some kind of enhancers. Enhancers are the chemical enhancers. They're the chemicals. How much you can enhance? How much you can enhance the permeation of the drug molecule? Generally, when you use for a research purpose, you can show like 30%, 30 times, 40 times, 50 times, or something. But most of the enhancers we cannot use in industry because we have to show the safety profile of that particular enhancer. Is it safe or something? At one point, people used the DMSO, dimethyl sulfoxide, very good enhancer, but unfortunately, it's not acceptable because of its toxic profile. So these enhancers within the limit, if you go to the FDA website, you can find out inactive ingredient. In the inactive ingredient, what are the inactive ingredients used for the skin patches, and what is the percentage you can use. Generally, you cannot use more than 5 to 10% of these enhancers, and how much they can increase, about 4 to 5 times. If my drug molecule by itself is penetrating 4 to 5 times lower, I can think about adding enhancer that can increase 4 to 5 times. But anything like 20 times, 30 times difference, from my blood level requirement, I cannot add. That's very difficult to make a patch with it. That's one of the limitations. So what enhancers? There are so much of chemical classification if you go through any book, but when it comes to the functional classification, there are skin retention enhancers and skin transport enhancers. Skin retention means they stay in the stratum corneum and they change the structure and they facilitate the drug permeation. How they change it? They wash off some of the lipids, or they change the lipid bilayer. There are so many mechanisms involved. The second one is skin transport. The enhancer will go to the other level. It permeates with the drug, along with the drug, and take the drug to the other side, like dipropylene glycol, ethanol. Ethanol is not much used, but in reservoir patches, they use little quantity. In skin retention enhancers, you can see isopropyl myristate, some of the lipid soluble, oleic acid, volyl alcohol. I think I gave some examples here: oleic acid, IPM, volyl alcohol, isopropyl myristate, isopropyl palmitate, DPG, levulinic acid. These are the most commonly used and most commonly accepted from the regulatory standpoint, and they have been used in different patches. If somebody used these different patches, you can easily justify it, and they are such a old molecules, it's easy to justify, and there's a toxicity profile available. If you're using a new molecule, you have to have the DMS drug master file that shows what is the toxicity profiles, and it's a big file that should be submitted to the FDA. So you don't have to do all these things when you use these enhancers with percentage less than 5% or 10%. And solubilizers, and again, most of the drug molecules are not soluble to the required level. You can add solubilizer. Povidone. Povidone is the most commonly used. There are different types: povidone K30, povidone K90. K30 is most commonly used. There is also cross povidone. It's not that much used, but the povidones are a little bit on the hydrophilic side. You cannot use this povidone with the polyisobutylene, but they go very well with the acrylics. They increase the solubility. Also, they prevent the crystallization. If you remember, I told that we try to take the maximum saturation of the drug in the patch, about 90 to 95%, to increase the drug permeation. Those 90 to 95% also problem in the sense that they will crystallize when you put on the stability. That is the problem. The crystallization is the problem. The crystallization will have impact on the drug permeation. So povidone will decrease the crystallization of the drug in the patch. Up to some crystallization is acceptable, up to 5 to 10%. However, you have to show that doesn't have any effect on variability, no irritation, or the permeation is okay and all that kind of stuff. Antioxidants, this is very typical for any drug molecules. BHD, tocopherols, ascorbyl palmitate, and tacfers is another one. Tacfers, the main function is to increase the adhesiveness. You put the acrylic, still it's not good. You can add some of the tacfers to increase the adhesiveness. They are like mineral oil, hydrocarbon resins, rosin esters. So these are some of the chemical standpoint. And this is what is the ideal excipient you're expecting. They are predictable, reproducible, should not cause toxic to the skin, these excipients. They are compatible with the API. They should be gross material. Gross means generally recognized as safe material that is classified in the FDA website. Cosmetically acceptable, inexpensive. This is something you can think of, and you can write down some more. I don't know. Okay. Membranes. What kind of membranes you can get? I said there are release liner. For a typical patch, it comes with the adhesive matrix. That's where we already seen there is adhesive and also the drug in there with the enhancers and some other excipients. The release liner is something you can buy from companies like 3M. There are not many, but the most common one used is polyester, with silicone. Then there are fluorosilicone and fluorocarbons. They are not much used, but they came into use just because of the polyester with silicone has a problem. As I mentioned before, this is the one used for the Datrana. Then because of the amine drug in silicone, in the presence of silicone, making interaction with the release liner, so it's very difficult to peel off. So many companies coming up with different adhesives, different release liners. So that is the trial this company made to come up with fluorosilicone. Still has a problem. Fluorocarbons, to avoid that interaction and to there is no interaction between the to avoid the interaction between the release liner and the adhesive matrix. The release liner should easily come off. Generally, put some kind of pressure, like I can discuss a little later. Then you have backing membranes. There are different backing membranes. They are coming like a 3 different layers. There's a tie-up layer in between. They come up like LDPE, PVC, LDPE, and you have LDPE at one side and EVA on the other side, EVA on the other side, HDP and EA on the other side, PUPET. Some of these non-oven polyester cloth, aluminum foil backing. When you are choosing the backing membrane, you have to put 2 important things into the consideration. The backing membrane decreases MVTR. MVTR is a moisture vapor transmission rate. That means the skin always loses some kind of moisture from the skin. Of course, when you have a physical exercise, you will have more, but always without, you can't see that, but there is always the skin is losing. That's how the thermoregulation works. When you put the patch decrease that water loss from the skin, thereby it will affect the thermoregulation and causes some problem to the skin. Especially there are some membranes, they don't allow water at all, which are called occlusive backings. These occlusive backings, what they do, they increase the skin permeation one way, and the second thing is that they cause irritation, especially when you are thinking about using a patch for 7 days. The occlusive backings are really difficult to do that. But there are some other, like non-oven polyester and cloth, they are breathable backings. Breathable backings means they allow the moisture to leave through the backing to some extent, and they don't cause much irritation. So depending on if you're going for 9-hour or 12-hour patch, you can go with the occlusive backing. There's no issue. But for 3.5 days, for 7 days, you have to think about better to use non-occlusive backing. And the second factor to be considered is the drug absorption. When you put the patch for a long time, the backing absorbs the drug. The release liner is very thinner. The chances of release liner absorbing the drug is very low compared to the backing membranes. They absorb the drug, and we have seen many times. Once they absorb the drug, there is a chance that the drug may be permeating. Somebody come in contact with the patch of the other person wearing it. There is a chances for the transmission of the drug to the other person. As I said before, this is a very problematic with some control drugs or abusive drugs, addictive drugs like fentanyl, kind of thing, or very potent drugs like clonidines and scopolamine, nicotine. It easily get into the other people's blood. FDA is emphasizing, especially for topical gels, not for the patches. There is a little bit of problem. For the gels, there is a big problem. If somebody is having the testosterone gel on the biceps and it's a transdermal, somebody come in contact, the loved ones come in contact with them, children or parents, whoever, there is a chance that that may pass on to the other person. So the backing membranes, coming back to the backing membranes, you have to consider about these backing membranes absorb the drug and drug. So therefore, the drug level in the patch decreases. Therefore, on the stability, you get less and less and less permeation from the time zero. And adhesive membranes are used for the reservoir patches. When your adhesive doesn't have very good adhesiveness, you can have adhesive membrane. This is another PSA. You just coat on the cast on the matrix. Control membranes, as I said, they are not very much used unless for something like a 7-day. The scalability is very difficult because you are going to go with the multi-layer patches. Not every company will have that kind of capability, and when it comes to the capability of the process, this is challenging. Manufacturing considerations. Manufacturing has, I think I just go with the manufacturing process followed by specific tests, not all the tests. With that, I will complete it. I thought that this is for 45 minutes. I think it's going more than that. If you guys don't have any time issues, and kindly bear with me, I will complete another 20 minutes, 15 minutes maybe. Mixing, you put all, probably I can show you pictures, that way you can understand. I will go back and forth in my slides. I can explain to you. This is not the one I want. This is the one. Okay, here is the mixing. Mixing, you put the adhesive. Hello?
Yes, sir. You can go ahead. You can go ahead.
Okay, the adhesive, as I said, the adhesive is a polymers. It's a silicone, acrylics, and the PIB polymers. So the mixing should be very gentle. They are thick. You should not use, they are the paddle mixers. You should not use very high shear mixers like homogenizer. Homogeneous mixes very fast, and it causes a vortex and picks up some of the air from the environment. And also the biggest disadvantage is that it cuts the polymers. We don't want to have polymers cut into monomers. Those monomers will have issues later on. And again, the mixing is challenging when it comes to the PIB, which is the thickest polymer. And you can add API enhancers, other excipients. Sometimes the order of adding the polymer, order of adding the raw materials is challenging. They will have impact later on in the casting process. The second step, once you have everything is mixed down here, this is actually a casting and heating zone and backing casting. Okay, here we always put the semi-solid or semi-liquid kind of preparation on this is the release layer. We put on the release layer, not on the backing membrane. We put on the release layer. Here is the drug coming from this kettle tank to the layer here, where the drug will be coated on the release membrane. You can set what is the thickness you want. That is again a lot of work need to be done before you come to that level. We say the word called coat weight. The coat weight is milligram per centimeter square. How much adhesive matrix milligram per centimeter square is called coat weight, and how much percentage of the drug you put in there. You have to mention all those come in the formulation, the formula, in order to have the formula. Then once it has a lot of solvents, mostly heptane, ethyl acetate, other solvents. Then the release layer with your the drug coated on that one, it will go into the heat zone. This is a tunnel. In the tunnel, this will be going moving at a slow speed. The tunnels are different lengths, like they are like 6 feet, 12 feet, 18 feet, 32 feet length. 6 feet is very small. They are used for the pilot purpose, but mostly the industrial purpose is like 12 feet, 18 feet. When it is passing here, there is a temperature created here. There are different temperature zones. The temperature, what will happen? The temperature will take off all the residual solvents and thicken the cast, thicken the polymer cast on the release liner. This is one of the challenging. Here is the thickness, and here is the challenging thing is that when you wanted to evaporate the residual solvents, you also lose some drug, and the process may create some excipients, and okay, you may lose the enhancers. We have seen this case. We have seen this many times. We lose the enhancer, you lose the drug. You don't get rid of the residual solvents to the compoundial limit, and you will have all these issues. But this is the best place to use your QBD because there are so many parameters. The speed of the tunnel, and you go with like 1 feet per second, 1 feet per minute, or 2 feet per minute. And what is the temperature you are creating? It's 80 degrees, 90 degrees. Very, very difficult. Some of the liquid drugs, you got to be very careful. The liquid drugs, they evaporate really fast. So what is the temperature you are creating? You will have some kind of preliminary experiments, and you come up with your operating parameters. I'm just talking about the QBD, how much you can apply QBD here. Say, for example, temperature, I just go with the two sides of the range, 60 to 90, one at 60 and one at 90. What is the tunnel speed? 1 feet per second versus 2 feet per second. And you apply the DOE, design of experiments, using your Minitab or some other software. And you can get the results. You put in your results in your Minitab, come up with your response surface plots or counter plots, whatever. Then you can create what is the design space. The design space is the most possible range. You get a product within the specification. The design space is very important because even if you alter in the future any of these parameters within the design space, you just don't need to refile with the FDA. You just inform them. We just change the parameters. So there's a lot of scope here to apply the QBD and design of experiments. And once you get this layer here with the release liner, this is the backing layer. You just get this one of the backing layer. Once that is done, you have to leave that one for in a bundle. It's a roll bundle. You have to leave that one for like a 2 weeks, 3 weeks, 5 weeks, depending upon the product. That's called curing. Curing means the polymerization takes place in the adhesive, and it gets toughened. The polymer layer becomes toughened and reaches to at one level. It's last stage. It won't go anymore after that. So we need to find out the time, how much it is taking. Is it taking like 2 weeks to complete the curing, 3 weeks to complete the curing, or 5 weeks to completing the curing? So you're pretty much you're leaving the bundle at the room temperature until that process completes. Then once that is done, after curing is done, this step is called slitting. Here, this is the bundle with the full patch, and it will be slit into layers and different segments, and these segments are rolled again here. That is the slitting part, and after the slitting, here is the cutting into pouch and packing here. This is the slit one. It is going here. It die cut into the patches, and it going here. Then it will be pouched here. All the pouches are cartoned here. The diagrams are schematic diagrams. They look very simple, but it's not so simple. Everywhere there is a chance You need to set up the parameter, the specs, specifications, how you come up with the specifications, how you justify them using the DOE, and how to come up with the design space in the QBD. So a lot of work need to be done there, but it looks very simple. Those are the considerations you have to take into the manufacturing. I'm just seeing anything I missed here. Okay, pretty much I think covered. It's misnomer to say covered because there's a lot of signs. I'm just going the skeleton, touching the skeleton. This pictures copied from this company called Lohmann. It's a German company. They do a contract manufacturing product. Okay, coming to the last topic, the specific tests we do for the transdermals. There are four specific tests. This is something more and more the regulatory agencies are asking for it. There are like four. One test is the peel tester. Peel tester means when you put the patch onto the skin, is it easy to peel when you're done with after 1 day or 2 days of treatment? Is it easy to peel, or it stick to the skin so bad when you peel it come off with your skin layers too, the stratum corneum? So that will be a issue. So you have to show that this patch is easy to peel. The biggest challenge here, these tests are accepted, adapted from the companies that makes the PSAs, pressure sensitive adhesives. None of them are done on the skin. We use a stainless steel plate. On the stainless steel plate, you put the patch. Here I see here is the stainless steel plate, and on the top of the stainless steel plate, you put the patch. And here is the lever, and you pull it. And when it pulls, the how much force is needed to pull this one, it will show here. This is the peel tester. You have to do for the patch, and also you have to do for the release liner. That is the recent requirement. Recent requirement means last 7-8 years, to show that the release liner is not tightly bound to the adhesive matrix. Peel tester, this for adhesion and release, we are using the stainless steel plate, and you have to do at 90 degrees and 180 degrees centigrade. How we pull this one? 90 for the patch and 180 for the release liner. You have two different pull forces, 2 pound and 5 pound. You have to put like this much of transround patch on the stainless steel plate. It's very interesting. There's a lot of question, a lot of discussion how these values correlate to the skin. There is no clear answer for all these things. So much of work done, and if I get a 200 value, how would really that correlate to the skin? A lot of work done, but there is nothing much. But what we do, what are the skin what are the patch we think is better? At least we choose like 2 to 3 patches, maybe more than that. We do the variability studies, placebo variability studies. When there is a drug, you should not do any variability studies. We take like 10 to 15 subjects. We ask them where it's supposed to go, below the navel or the other biceps or whatever, with the body hair, without body hair, or maybe sometimes shave off, and take for 7 days or 3.5 days, whatever the time period, and see how many patches are falling off. The patch that stick very well, those values, those patches, we come back and see what are the values on this machine, and we take those values, and we have to put the spec also for that one. The specs should be tight. Sometimes companies want to take the advantage and they go for a little wider specs, but there is always a pushback from the industry to make the specs tighter. This is the peel tester. So this gives a what is the high force, low force, and what is the average, and all this. the values comes in grams, kilograms, whatever you can make those changes. Shear tester is another one. What is the cohesive force within the patch? The cohesive force is very important because if it's like if it's like a semi-solid or anything, when you put the patch, the adhesive will seep horizontally on your skin, and shear, that is one of the biggest problem with the if there is no proper shear. Shear is mainly talking about the within the patch. Adhesive is talking about patch to the skin, whereas the shear is talking about within the patch. How we measure it? We just hang some weight, about 250 gram or 500 gram weight from a patch attached to a reader. How much time it takes for the weight to fall off? That is the measurement. Okay, this is measure the cohesive strength of the patch. We use about 200 to 500 grams, and we measure how much time it takes to fall off for the shear strength. That is noted in minutes. Again, I said it's a no skin technique. Those values we come back and put the specs and everything. Okay, this is the tag. Tag is another one for adhesive testing. You just touch the patch like a fraction of a second, like 1 second, and when you remove this one, touch the patch for a second or so. Then when it is coming off to the down, how much pressure it is needed to pull out from the patch. That shows the adhesion. There are different kind of tags, probe tag, rolling ball. I see when I was working in Tiger Balm patch in Singapore, they were using a rolling ball. Rolling ball is something you have a slanted patch like this on a plate, and with a small ball, you roll on it, and where it stops on the patch, that shows the adhesion. If it falls off without attaching anywhere, that is considered to be very low adhesive. It stops very top of the patch, that is considered to be very good adhesive. But in USA, that's not used much in rolling ball. This is a measure adhesive. These are the three main testing for adhesion. This is the last one I'm going to go with, and with that one, I'm done with it. Cold flow. Cold flow is another issue with the, especially patches when we put on the stability. What happens? The PSA is like a semi-solid, semi-liquid kind of thing. With the time, it seeps. See, this is the starting patch, and you see it seeps here. It is seeping here. That means the adhesive is coming to the sides. What is the disadvantage with this one? Because you are not getting the complete drug when you put on to the skin. The second thing is that when you put onto the skin also, there will be a black ring. If you put for 7 days, there will be a black ring. And when you take off the patch or somebody comes into contact, the drug, whatever the drug that is in the outer layer, which is the cold flow we say, that will be transferred into the other people. So the cold flow should be tested and should be within specific certain limits. And FDA is very, very asking all this information. There are different ways of measuring it. One is the gravimetric. You put some kind of weight on them, and how much seeping in the patch you can see, and weigh how much is coming out of that one. That's the weight. There's a gravimetric analysis. Imaging analysis is that you take a three-dimensional microscopic, and you find out, you can find out what is the weight that has come out of this patch. There is a cold flow. Those are specific tests that are done in addition to your regular dissolution testing and other testing, the assay, uniformity, and all that kind of testing. The skin flux, in vitro skin flux, so far is not asked by any agency, but however, The reason being that there's a big bio variability because of the biological tissue. The variability is very high. The results variability, you can say anywhere from 90 to 60 to 100%. For the last 30 years, so much of in vitro skin work done, but not accepted by the regulatory agencies. However, we are getting more and more experience, knowledge acquired on this skin permeation studies. Regulatory agencies are accepting the data. EMA is accepting the data, and FDA is actively asking for this data these days. Especially, you can use this data when you make minor changes in your product, like change a non-functional excipient or one excipient with a similar excipient. You don't have to go for the clinical studies. You just do the in vitro permeation studies, and these days they're gradually accepting it. And also heat PK studies. If somebody, heat is it is in-use study. In-use study means somebody exercise with a patch on him, or go to some swimming, or what will happen during that time. That kind of studies they are accepting. For generics, they are accepting the skin permeation studies, in vitro skin permeation studies. So the importance of the skin in vitro studies also increasing as a replacement to clinical studies in some minor cases, in minor changes. So far, it took longer than what I was supposed to take, but however, I thought that this information hopefully useful for you guys. I touched from the skin point of view. I touched the patch point of view. I touched the manufacturing standpoint. I touched the specific test. That is not the entire thing. There's a lot more. This is a big ocean, a lot of work, but I try to touch the skeletons. With that, I stop here, and I also thank for giving this opportunity to share what are the small information I have with you guys, and you can ask some questions if you have.
Thank you, sir. Thank you very much. Maybe Professor Nagaraj, sir.
Please. Yeah, I am online.
Yes, sir. You can go ahead with the
Can you hear me?
questions.
Yes.
Yes, sir.
Yeah. It is a very good lecture by Dr. Harnath. He has covered every aspect of transdermal delivery, and also very important is for your information, sir. 3,400 people have watched this on the YouTube and on the Zoom. Amongst them, 2,200 are the faculty, teachers, and 1,200 are the B.Pharm, M.Pharm, and PhD students who have watched your presentation for the last 2 hours. So thank you very much for your beautiful narration, where everyone can understand. We had ample number of questions, but however, our committee has screened, and we are going to ask only 2 or 3 questions to save the time. First, number one, since this particular area of research is not sometimes very difficult to do it in our academic platforms. So what do you suggest these type of projects when you take it to the laboratory of the colleges? What should be the crucial areas where we need to concentrate?
I don't know about, but I see a lot of work going on in the United States. A lot of work is going on, as I said, from different perspectives, from the skin point, skin mechanism, how skin affects, what is the interaction of the drug and the skin and the enhancers on the skin, and how to make the patches. There is so much of research going on in Europe and USA. One of the challenges I see for this type of dosage form, the first product came in 1980s, and now in 30-40 years we see only 15 patches in the market. That says it has a lot of difficulty to develop this kind of dosage form. For the passive patches, most of the low-lying fruits already taken. Most of the easily developable molecules have been taken, and we screen a lot of molecules in about 100 to 200 molecules. We screen them, and many of them are not successful from the technical point of view, or business, or clinical, no justification. However, there is a switch going on from passive dosage form to the active dosage form, using the iontophoresis and microneedles. Microneedles, they make a very minute pokes on the stratum corneum and increase the permeation, especially that is used for the larger molecules. And iontophoresis is a kind of small electricity created on the skin to push the drug across the skin. There are a couple of molecules at the phase 3. One is sumatriptan for microneedles is in the phase 3, and there's another patch, JQT, that was withdrawn because of the shock to the skin by the iontophoresis. Yeah, I think the academic research now should switch from passive to the active. That is where a lot of potential research in the future. And they are not really difficult. You can get derma rollers or something you can easily get, and iontophoresis is not also a very complicated thing for research lab level. You guys, I mean, the future will be the active transdermal. That will be one area that can be focused. And most of the equipment used at the lab level is not that expensive. If you want to make a gel, not necessarily patch. You can use with a paddle mixer. They're not expensive, and also there will be a gap set. Gap set means how much thick you need to get the patch laminate. Those gap sets are available in the market. They are also cheap. They're not very expensive. And what I'm trying to say that you can use mostly on the active dosage form, active transdermal. That is a lot of work is going on in the active dosage form, using the biomolecules for the active. But for the passive, a lot of work already has been done, and many molecules are not suitable for small molecules are not suitable for transdermal. All the easy ones already in the market. What are the 15-20 molecules? So I suggest that academics should more focus on the active transdermal, and they're not cheap, they're not that expensive. Those small stuff you can buy easily.
Thank
I don't know
you,
whether
sir.
I answered your question properly, but
yes, it has reached. We have got. Yes, sir. The second one is, what about the application of this technology for the natural products, especially the plant extracts and herbal extracts and all that? Can we make a transdermal patch out of it?
That's a good question. We need to have a lot of data about those plant extracts. My understanding is that these plant extracts are generally not that potent. The dose will be higher, 500 milligram to 1000 milligram or whatever. As I said, there's a limitation for the transdermal patches, passive transdermal patches. It should be less than 10 milligram. However, you can think about making a bigger patch if you have a little bit higher dose, up to 50 milligram. But there are so many patches for the local skin purpose, not for systemic purpose. There are so many pain relieving patches, but I never come across the plant extracts. But I feel that is the reason. One reason being that the dose may be higher. Second, the processing may be difficult because all of them need to dissolve in the adhesive matrix. Most of the adhesive matrix are very oily in nature. How much they can go into that adhesive matrix mix when you go for higher quantity of the API, and And they are the mix of molecules, the natural products. Generally, you don't get like one single mix, and you don't know which one you are going to measure in the plasma, which particular constant you are looking for in plasma. We don't know. And what I'm saying, having said all that, it's difficult for plant extracts or because of their high doses. But you can think about using for the cosmetic purpose or some in terms of pain relieving or topical purpose, so topical therapeutic efficacy or cosmetic purpose. Those things can be explored.
Thank you. Sir, what is the status of transdermal insulin preparations? Anybody is working on it, or still it is not coming to reality? What may be the challenges, in our opinion, where it is stopping us to move further?
Which one you said? Sorry, which molecule?
Insulin.
Oh, insulin. Oh, okay. Insulin. Yeah, a lot of work is going on insulin using the passive patches. Is not going to go because of the molecule size is very high.
Yep.
And a lot of work is going on working with the active systems, active transdermal systems, using the microneedles, especially. As I said, microneedles cause a small poke, very tiny pokes on the stratum corneum, and the bigger molecules, the biomolecules, can go through it. And right now they're working on a vaccine, flu vaccine, which is in phase 3, if I'm not wrong. That's a bigger molecule. And a lot of work is going on, not only for the insulin alone, but insulin-related molecules like exenatide and other stuff. Using other ones like PTH, parathyroid hormone. They're all big molecules. Actually, we did some kind of exploration in the direction using the microneedles. A lot of work is going on, but I don't know any product in any clinical at this point. Is still at the exploration level, even still at the feasibility level. Hopefully, if once the microneedle system establishes itself as a promising way of releasing the drugs into the blood, hopefully at one point we will see some of the biological molecules that includes the insulin, exenatide, or some other biomolecules available as transdermal dosage form active. One of the disadvantages is that you will have every patient should be dispensed with an applicator. Applicator is some kind of device you have to put onto the skin and make a small holes, and after removing that one, you have to put the patch. Or sometimes the patches themselves have microneedles you can just press onto the skin. For the short acting is okay, because the long acting there is an issue that the skin itself has a very good healing property. It will heal by itself within 12 hours, and you cannot deliver the drug continuously. But if you are looking like a vaccine or like a one-time shot for insulin or PTH hormones or small biological molecules, hopefully one day, I don't know, maybe another 5 to 10 years, we could be able to see some kind of dosage forms. May not be necessarily for insulin, but we will see for biologicals at one point for sure. That's what I'm thinking.
Maybe the last question, sir. How is the possibility of delivering vitamins and iron sort of things through transdermal?
Vitamins, I see some OTC molecules. Vitamins, again, the dosage form, the dose is very high. The same thing, there are some mild soluble vitamins and water soluble vitamins. Water soluble vitamins, I don't see much scope because they're water soluble. They cannot penetrate through the skin in the passive patch. You have to look for some kind of active patch, and the biggest disadvantage is that their dose is very high. Oil soluble ADEK, they are again could be explorable, but again the dosage form is very high. At one point, we explored vitamin D, cholecalciferol, along with the PTH, but vitamin dose was really high, like I don't really remember, like 500 milligram, which that is expressed in units. So I don't see, unless the requirement is very, very low. I don't see vitamins could be possible in transdermal. Second thing is that vitamins you are giving through the water route, they are very good. You can get whatever the quantity. I don't see any business or clinical justification there, because the transdermals are expensive for sure, especially when you are going for the active. You have to supply that applicator also along with it. And you put a patch, the manufacturing process is expensive. And when you are getting a cheap water route dosage form for the vitamins, I don't see any clinical or business justification, and also the technical feasibility is questionable.
Thank you, sir. Even though we have many questions pouring in, I really don't discourage the participants to get the answers from a eminent speaker like you, sir. At the end of the session, at the end of the program, we'll have the panel discussion. So most of the important questions will be taken up there, and we'll be able to answer those questions. So for time being, I'll hand over the stage to mic to Dr. Satish for the further proceedings.
Thank you very much, sir. Thank you, Dr. Harnath, sir, and Professor Nagaraj Garu. Because we are running late by 40 minutes, maybe we can take up few more questions during the last day session, with your permission, sir.
Can you hear me?
Yes, sir.
Yeah.
Yes, sir.
Okay. Yeah, I know. I realized it. I'm late about 45 minutes. I was thinking about 45 minutes, but unfortunately it went little over the given time, mainly because so much of transdermal stuff there. I thought that I didn't do complete justification there, but whatever, I think I could able to give whatever the information that is sufficient for the students. Yeah, I don't know whether I would. Only weekend is the only one I can take those questions. The one you're planning about the panel discussion. Weekdays, I'm not available. If it's like a Saturday or Sunday, I'm more than happy to help you, help the students.
Thank you, sir. Thank you, sir. Thank you very much. We understand the value of your time, and we didn't want to disturb you because you want to complete the content as you were from the beginning. That's why we didn't disturb you. And there are many people who were eagerly listening to your presentation. So we want to give them that complete information. So it's not a few minutes more. It's not an issue about it. We'll be very happy if we can be there for the panel discussion. On behalf of our college and on behalf of the organizing committee, we wholeheartedly thank you for your excellent presentation as well as contribution, sir. Thank you very much.
Thank you, and good luck to the students and good luck to everyone.
Thank you very much, sir. And thanks to all the panelists. Thank you.